thank you anyways. A few clarifications before I start. As you can see, and as you may notice, my talk has had a little adjustment in the title, because if I would intend to talk about the changing phase of human evolution, we would need maybe a whole semester for it. So I will talk only about the changing praxis in the study of human evolution during the 21st century. I will read my talk because my English right now is a little rusty, so I wish you forgive me for that. And also I want to thank especially Ana Barahona for inviting me to this event. Also, I want to thank Carmen Adame for all the organization and the logistics of the activities and so on. And well, thank you for being here. Okay. So human evolution is a fascinating issue, both for its conceptual and methodological principles, as well as for its philosophical and epistemological basis. It is also an interesting topic because of the way in which its postulates have evolved throughout its history. In this presentation, I will address the changing praxis in the study of human evolution during the last 20 years as an outcome of the publication of the Human Genome Sequence. This is an important thing to say. Right now, we are 21 years ago from that publication, and its implications within the community of scholars focus on the study of human evolution. My starting point is the assumption that the possibility of reconstructing life histories from dead people or bodies is one of the main aims of physical or biological anthropology. Anthropologists achieve this goal through the analysis of human remains and the collection of data that can give light of such aspects as the age at death, sex, ancestry, stature, pathological conditions, traumatic injuries, and so on. The methods applied focus on recently dead people, as well as on archaeological and paleoanthropological remains. With regard to the archaeological remains, the analysis can provide relevant information not only about the single individual, but also about some characteristics of the biosocial and biocultural underpinnings of the community where the individual thrive. It depends on how many skeletons you can recover for having a more broad perspective of these issues. For instance, information related to demography, genealogical relationships, patterns of nutrition, migrations, among others, can be inferred through the study of these ancient human remains. Nevertheless, fossil evidence is frequently much more scarce and fragmentary, which makes it more difficult to extract accurate information from it. Notwithstanding these circumstances, morphologists and paleogenomicists have pushed their research work in order to advance the knowledge and information that can be collected from these ancient human remains. In this quest, there is a theoretical, methodological, and conceptual dispute among geometric morphometrics in anthropology and molecular biology-oriented studies, mainly the recovery, extraction, and sequencing of ancient DNA. This is DNA recovered from ancient bones, remote temporary bones. Well, the next one, please. Here I will develop two main points that delve into this transdisciplinary relationship. First, I address the ongoing shifting process from a more morphology-based theoretical account to a more molecular biology-oriented perspective of the human evolutionary process. To do this, I contrast how it is that geometric morphometrics and paleogenomics approach their object of study, the human fossil record. My next point will call for a specific example, the hominin fossil. The hominin fossil evidence from Denisova cave. aiming to illustrate in a more conspicuous fashion the epistemological tension and the scientific practice shift among these different ways of analyzing human fossil evidence. Finally, I close my presentation with a critical perspective that focuses on the argument of the enormous pragmatic importance of an accurate understanding of the human evolutionary process that is rooted in the need to integrate data and conclusions that both perspectives, paleogenomics and geometric morphometrics, and many more, can produce rather than pondering one over the other. So I will end my talk with some non-concluding remarks. Next one, please. What can bones tell us about the evolution of our species? First, we must be aware that we will never know exactly the intricate details of early human evolution, among other things because of the sparse and fragmentary evidence we biological anthropologists work with. However, this is also one of the most highlighting aspects of our discipline, one that makes it attractive and of course controversial. Therefore, human evolutionary process must be observed from a scientific perspective, one that manages and overcomes the harsh characteristics of the hominid fossil record. As anthropologists, we focus on the recovery, the next please, the recovery, conservation and analysis of ancient human remains as a means to gain knowledge on a diverse array of aspects related to nutrition, locomotion, social organization, ecological adaptation, and so on. On a basic level, and from a classical perspective, morphology, mainly size and shape of hominid bones and teeth, can reveal fundamental issues related to their ecological adaptation strategies. For instance, how they walk, how did they eat, how did they mate, etc. For example, related to the nutritional characteristics, we have the emblematic case of Paranthropus. The next, please. A hominin genus with very large teeth in mandible, particularly their molar teeth. The explanation of the morphological characteristics of this species has delved into the biomechanics of chewing. In other words, Paranthropus shows this prominent masticatory apparatus in order to deal with the large amount of food they consume that were lower in quality and tough, hard, or abrasive to consume. However, the question persists. How do morphologists recover information from such kind of evidence and translate it into a coherent narrative of the human evolutionary process? The history of this line of inquiry goes back to the 17th century. However, it is not my purpose to deepen here into a historiographic account of this issue. Nevertheless, it is worth referring Johann Friedrich Blumenbach as one of the first that considered the possibility of organizing human populations into broad clusters that map into geographical areas of the planet, which in turn represented typological, essentialist characteristics of their inhabitants. Of course, you can see the screen, the hierarchical organization of the human types, right? So he offered a classification of human races, claiming 5 of them: Caucasian, Mongolian, Malayan, Ethiopian, and American. This was, of course, a racist and racialist, as well as hierarchical and reductionist fashion in which to arrange human diversity. It is clear these assumptions do not follow how human variation is distributed between and within populations throughout the world. However, these ideas were important for the consolidation of physical anthropology. On this basis, and throughout the proposition of novel tools and concepts, morphological studies changed and progressed, and they have tried to get rid of its racialist assumptions, not always been able to do it. Thus, contemporary research on this line has had dramatic conceptual, methodological, and theoretical changes. For example, the range and detail of research questions has increased, involving aspects such as the possibility to determine to what species an osteological specimen belongs. Also, if sexual dimorphism in school morphology is or not related to sex-specific food procurement roles. Moreover, the phenotypic differences that arise during speciation are due to genomic reorganization, or can they be explained by simple allometric shifts? Following this line, it is feasible to state that geometric morphometrics is a technique particularly suited to test such questions and hypotheses because of the sophistication with which it can be used to measure phenotypic variation. Geometric morphometrics, in sum, is the analysis of Cartesian geometric coordinates of morphological structure rather than linear, aerial, or volumetric variables. For instance, when analyzing the evolution of the human brain, geometric morphometrics can yield accurate information about small shape differences between different species, brains, and skull morphologies. On this basis, Neubauer and his team developed a methodology that consisted in the analysis of endocasts. This is the internal part of the skulls of chimpanzees and modern humans to gain knowledge of brain growth and the adult shape of the skull among these taxa. They did it using three-dimensional landmarks and semi-landmarks, and they found that there is an early globularization phase in the development of the human brain, which does not occur in chimpanzees. This ontogenetic process facilitates the expression of parietal and cerebellar areas. In addition, Gons et al., using the same methodological framework, compared and analyzed the shape of the brain in modern humans and Neanderthals. They were not able to identify the globularization phase in the Neanderthal case, which forced them to conclude that Neanderthal brain growth is more similar to chimpanzee brain growth than it is to humans. This is an astonishing conclusion, and one of the many reasons that makes geometric morphometrics in anthropology an important tool for the study of morphological evolution in the hominid lineage. However, and this is very important, morphology cannot be directly translated into behavior, which is the ultimate goal of human evolution research. Not only give light of which species one given fossil belongs to, but also, and overall, how did that individual behave in a social context, and also in interaction with other species, of course. My critique here focuses on the problematic of extrapolating bull morphology evolution into mathematical data in order to achieve some understanding of ancient hominid evolution. It seems to me that this reductionist strategy is not able to capture the whole complexity of morphological evolution and its effects in hominids. Of course, it's obvious. Nevertheless, it helps to organize the human fossil record in order to detect regularities and discontinuities. In any case, a preliminary conclusion is that geometric morphometrics is a necessary tool for studying the hominization process, but not sufficient for a complete explanation of it. Okay. Well, what can genomes reveal about human evolution? Now I will talk about the other methodology for approximating fossil evidence and extracting potential data for its interpretation. So, recent developments in molecular biology-oriented studies regarding ancient DNA from human remains have brought into the contemporary discussions within archaeologists, bioanthropologists, and geneticists a set of disputes, tensions, and collaborations that need to be analyzed in practical and epistemological terms. Among other things, aDNA research, ancient DNA research, has moved from mitochondrial sequencing to become a fully genomic discipline that can contribute to our conception of human evolutionary process. There are areas of aDNA research that provide useful information regarding human adaptation and disease. So the potential of the studies of aDNA surpasses, of course, the focus of human evolution. However, molecular anthropology can be characterized following Bastian Yáñez, as a complementary approach that exploits the genetic diversity of contemporary human groups to understand their history and genetic makeup. These studies have sparked a heated debate on the contribution that molecular biology-oriented studies might make to solving long-standing questions in archaeology, biological anthropology, and other anthropological research areas. This recent prominence of aDNA in restructuring visions and versions of the past does not necessarily entail that the science of aDNA is somehow epistemically privileged over other archaeological and bioanthropological analysis, fundamentally geometric morphometrics, but some other disciplines also. However, next, there is a sense of significance and realness surrounding assertions derived from aDNA studies that give them scientific weight, and this is not exclusively drawn from the actions of the community of aDNA researchers. In the image, you can see the machines that help researchers to extract information, big data, while they are sleeping in their homes. These robots are working and extracting information, which contrasts in a very impactful way with the other methodology in which we have to measure the bones. And this, of course, brings some problematics also, the human error, of course. Okay, DNA then, or I must say also that technology is, of course, a significant variable in this scientific inquiry. Maybe in everyone, but in this one, it's really, really important. DNA, regardless of what form or type it is, mitochondrial, sedimental, DNA, nuclear DNA, etc., carries with it an assumed explanatory significance that might indeed be hitting above its weight class in terms of essentialist and materialist biologized explanations of what and why things are. In fact, one might go so far as to suggest we are in the midst of an aDNA frenzy or a gold rush of aDNA, which reflects the shifting process of methodological and conceptual changes in the study of human evolution in the last 20 years. In addition, over the past 2 decades, the uptick in the ability to extract DNA samples from bones that are many millennia old and the radical advances in the ability to amplify those sequences and analyze them has added substantially to our data sets on the human past, especially in regard to the complex movements, migrations, and demographic dynamics of the terminal Pleistocene and the Holocene. aDNA analysis have also forced substantive revision of our understanding of what constitutes Homo sapiens genomically, reinforce the reality that genetic diversity is not only a contemporary phenomenon, and reveal that many assumptions about phyletic histories of Pleistocene Homo likely overstated boundaries and distinctions between populations and taxa in our genus. This is a very interesting discussion right now happening in the realm of the human evolutionary studies. In short, aDNA research has revolutionized the toolkit and analysis of the human story. However, and I want to underline this, however, this increased technological and analytic capacity has not been separate from the problematic histories and practices in the fields of human evolution, biological anthropology, and archaeology. Next, please. These problematic aspects of aDNA research focus on the use and abuse of samples, ethical, moral, and legal obligations, formal recognition of and consultation with all stakeholders, and so on. Of course, these critiques are more focused on an ethical and and in the way the human remains are managed. Nevertheless, there are also relevant aspects related with the methodological and conceptual changes in the study of human fossil evidence that are worth mentioning from a critical perspective. Two of them, mainly the pace of the practice and the reductionist perspective of molecular studies. For instance, in February 2021 was the commemoration, as I already said, of the 20th anniversary of the first publication of the human genome sequence. Next, please. It is important to point out this. Prior to 2010, no one had sequenced a single complete genome from an ancient human, while today that tally tops 6,000. A single paper published in Nature in December 2021 sequenced genomes from 793 individuals, requiring, of course, collaborations among hundreds of scientists. This gives us an accurate idea of the accelerated pace with which paleogenomicists are working and reveals the highlighting stance that aDNA studies have occupied in the last 20 years. It means, as I was telling in the beginning, this epistemological primacy that is in dispute among morphometrists and paleogenomicists is being, right now, the paleogenomicists are leading this race or this struggle. Again, one can raise a set of critiques towards the way in which aDNA studies are advancing and monopolizing part of the human evolutionary scientific endeavor. Nevertheless, in this final comment of this section, I will focus my critique in the ultra-reductionistic nature of paleogenomic research. As I already mentioned, geometric morphometrics is a suitable methodology for the analysis of bone morphology evolution. However, it is reductionistic in its results and not sufficient for giving light of the multifactorial aspects related with the human evolutionary processes. Following this line, paleogenomics, in my perspective, is even more reductionistic than geometric morphometrics is. However, this technique is useful because with the correct anthropological and archaeological context, molecular data can yield important complementary information. Nevertheless, isolated genomic data has not useful potential. And this is something that geneticists are not always aware of. In the same vein as with geometric morphometrics, aDNA analysis are necessary studies when possible, because not always it is possible, but they are, again, not sufficient to explain human evolutionary history. I will go now for an example to illustrate this point. Next, please. The hominin from Denisova. In 2008, paleoanthropologists excavated from the Denisova cave in the Altai Mountains in southern Siberia a set of hominin fossil bones with an astonishing high degree of conservation. 2 years later, 2010, Johannes Krause and his research team reported a complete mitochondrial DNA sequence retrieved from these human remains. The results showed a confusing account that indicated an interbreeding event of modern humans with an unknown species from the fossil record. Furthermore, these results suggested a hominin migration out of Africa, distinct from that of the ancestors of Neanderthals and modern humans, of the last common ancestor of Neanderthals and modern humans. On this ground, this was the first time the history of paleoanthropological research, this was the first time in the history of paleoanthropological research that a new species was inferred not from the morphological analysis of the fossil evidence, but on the grounds of molecular anthropological analysis. This is the highlight of my talk, of course. I'm sorry for pointing out. This is why the hominin from Denisova is not called Homo denisovanensis, because there is no paleontological remains that characterize this new taxon. In other words, we are talking here about a genomical instead of a taxonomic species, we have here a genomically defined species. Another conclusion from Krause and colleagues was that the Denisova hominin lived close in time and space with Neanderthals as well as with modern humans. Another surprising aspect that is worth to mention is that the bone fragment that yielded such intriguing results, that's fine. Is that the bone fragment that yielded such intriguing results was exclusively from a finger bone. That is why I have already said there is no sufficient fossil evidence of this species to characterize it physically. In the screen, you can see the teeny tiny bone from where this information was extracted. So in that moment, the only physical evidence of the Denisova hominin was the finger bone. Nevertheless, and this is also really interesting, researchers had the full genome of this until that moment on unknown taxon. This is an important example of the methodological and conceptual changes that have been happening in paleoanthropological research in the last 10 to 15 years. Sorry, there are until now a few fossils recovered associated with Denisovans, some teeth, a couple pieces of large bones, a mandible, and precisely 2 days ago, it was published the morphological description of a molar associated to the Denisovan fossil record based in its morphological characteristics in comparison with the mandible previously recovered. It is ironic, however, that for a definitive conclusion in terms of its belonging to any taxonomic lineage, it is necessary to analyze these remains, the molecular constitution of these remains. In other words, the morphological insights related with this molar piece are not sufficient to conclude if the fossil belongs or not to a Denisovan. In addition, the ancient DNA analysis is necessary, of course. However, it might be not possible to retrieve molecular information of this piece due to its conservation status and contamination, DNA contamination, and so on. So we are kind of in a problem there. So this last assertion leads me to my not concluding remarks. Well, I am sure that this will be a textbook case of the conceptual change in the history of sciences someday, particularly in paleoanthropological research. My focus on this analytical description is oriented not only from the perspective of a biological anthropologist, but also from that of a historian of science. In this sense, it is important to highlight the difficulty of deploying this analysis when the historical process in question is an ongoing one. In other words, from the perspective of the history of science, there is not enough distance, if you will, temporal distance, to establish an accurate historiographical perspective, since we are making and writing history at the same time. As I said in the beginning of my talk, there is an enormous pragmatic importance of an accurate understanding of the human evolutionary process that is rooted in the need to integrate data and conclusions that both perspectives, paleogenomics and geometric morphometrics, and many more can produce, rather than pondering one over the other. A second insight from this analysis claims a more geopolitical or sociopolitical perspective of the study and overall the praxis of human evolution. As it is the case in many more disciplines, core periphery or global north and south interactions characterize international scientific collaborations, perpetuating and reproducing asymmetries and inequities. In this context, the rapid pace at which ancient DNA research is advancing is forcing scientists in the field, from archaeologists to geneticists, to confront the ethical implications of their work. Sampling aDNA often leads to the irreversible destruction of human remains, for example, and might generate sensitive information about living people. Who decides then what research should move forward? And who has ownership over the products of that work? One certainty, if not a conclusion, is that the next generation of scientists will need better training to bridge the gap between archaeology, anthropology, and genetics, and be able to catch up with the methodological and conceptual changes new discoveries and new technological possibilities will bring. Thank you very much.